Super. Hey, welcome everyone. Welcome to another webinar by the Choroideremia Research Foundation. My name is Beth Voss, and I am the Director of Operations. Today, we are going to be talking about something really important today that many of you may have heard or have learned about. It's called endpoints. Endpoints are something that is a part of clinical trials that we have gone through at the foundation several times. So today we are very fortunate to have Dr. Amandeep Josan here, and who works with Dr. Robert MacLaren in Oxford. So we're super glad to have you here. Thank you so much. I think your volume you need to turn up your volume. Let's try that.
Can you hear me?
There we go. Perfect. Perfect. So before we get started, I just wanted to tell everyone we are going to take questions throughout. I'll try to be judicious, and when I ask them, we can take them, of course, at the end, or we can take them during the discussion that we're having today. So go ahead, I'll monitor the question and answer or the chat feature and pose them to Dr. Josan. And we want to thank our sponsor, 4DMT, for helping put these on. So how are things over in England?
Lovely, thank you. It's cold. We're coming into winter, I guess, same as you guys. But it's quite pleasant at the moment. Autumn leaves.
Oh, they are beautiful, that's for sure.
But
Listen, I'm going to go ahead and minimize myself and mute, and please go ahead and begin.
Thank you very much. I'll just share my screen.
There we go.
Right. Okay. Thank you very much, Beth, and thank you to everyone at the Choroideremia Research Foundation for the invitation. So I'm going to be talking about functional measurements in choroideremia and as Beth said, endpoints. As you probably know, endpoints have become one of the biggest topics in pretty much all of inherited retinal diseases, with gene therapies and all sorts of therapies. Is how do you measure whether it's been effective or not? People think of a potential cure as maybe just reversing a condition, but they rarely do that. They usually either slow the condition down or halt it, hopefully, or at the very best case scenario, can reverse it, but not to a huge degree. We're talking about mainly halting a progression. And that's quite difficult to work out how that's happened with the measures we have. So I'm going to talk a little bit more about that. Just as an introduction, my name, as I said, is Amandeep Josan. I'm a research optometrist at the Oxford Eye Hospital, working with Professor MacLaren as part of his lab group and the eye research group at Oxford since 2019. I started at the very end of 2019, about 2 months before COVID hit. So my uniform was pretty much this when I first started working from home, as all clinical trials pretty much halted at that point. And so we stayed at home. But that was actually quite good for me in terms of I learned some new skills. I took to data analysis. We had a lot of time, so I learned some new skills in data analytics. And that proved to be quite useful now. Once COVID was over, we got clinical trials back up and running. My main core job is to monitor patients that are enrolled on clinical trials, performing functional tests, like vision testing. And then when we don't have patients in, it's trying to research these functional tests, things like visual acuities and the perimetry, and trying to work out what the optimal tests are and how to perform them to get the best results. So I'm sure Professor MacLaren's no stranger to this group. He's also wears a lot of hats. He's an expert geneticist, and also one of the leading retinal surgeons in the world. Just when I Googled, I was interested to see how much he's written on choroideremia. And when I Googled it, it came up as a lot more than I realized. Came up as over 100 articles where he's authored on choroideremia. In so many, in fact, even Google gave up counting, and it wrote about 106. But he's obviously a world-leading expert, and is and a large part of why I've got into this is because of his guidance. So, as we all know in this group, choroideremia is a progressive disease caused by mutations in the CHM gene, which is responsible for encoding a protein, a specific protein called REP1. And that's crucial for cellular trafficking, basically the function of photoreceptors in the retina. It's an X-linked disease, affecting approximately 1 in 50,000 males, but the numbers vary depending on what surveys you look at. Generally, again, as you all know, presents with night vision issues, peripheral vision issues, and much later visual acuity decline. And that's quite crucial as to what I'm going to be talking about next, is the visual acuity aspect. When I look at clinicaltrials.gov, it comes up as clinical trials in choroideremia come up about 27 on here. And in Oxford, we were involved with some large choroideremia trials, as I'm sure you all know. The first one was an observational trial called NIGHT. And he was looking at the natural history of choroideremia, how things progress naturally, so we know what to look for in an interventional trial, what to monitor. So the first phase 1, which is always a safety stage of clinical trials, for GT1, followed by the regenerate phase 2, finally the STAR, which was the phase 3. And the primary outcome measure, the endpoint that we were looking for in phase 2 and phase 3, was the visual acuity, comparing before versus after the treatment. And you've probably seen, this was the recent-ish, last year was published, the results of the phase 3 STAR trial. And at the bottom here, I've highlighted, and as we know, it didn't meet the primary endpoint of a 15-letter gain from baseline. And I've highlighted at the bottom here some of the things that Professor MacLaren pointed out, was that the study for choroideremia in future needed optimization for the entry criteria. So choosing the best patients that are likely to get the 15-letter gain or some sort of gain. The surgical techniques, but crucially here, optimization of the clinical endpoints. So we were quite aware, or Professor MacLaren was very aware early on, that the visual acuity wasn't the best measure, but this was something that the FDA really insisted on. So measurements, measurements in clinical trials are broadly split into 2 or 3 categories. One is structural, the other functional, and more recently, there's patient-reported questionnaires, that sort of thing. But I'm going to focus on the 2, structural and functional. So structural are things like the retinal photography, the color photography, the OCT images, and the autofluorescence images. So these are just some of the examples that are shown here. And they're generally all about photography. The functional ones are more ones that the patient has to respond, press a button, read a chart out loud, something where the patient has to do something. So this is just a fairly short list of the options here, but the list goes on and on. There are countless functional tests you can do. So the FDA and the EMA, which is the European equivalent, generally don't like structural endpoints. They don't like clinical trials where you say we're going to look at the pictures and see if it's better after treatment versus before treatment. They have generally and historically gone for functional measures. So improvements in visual acuity, improvements in visual field, things like that, which is it's kind of understandable in terms of the functional aspect is what really matters to the patient, the fact that they can see better, their experiences have improved. But they are notoriously difficult to measure in an accurate way, a really highly defined way. Whereas structural measures, you can clearly see if there's a change there on a picture. You've got evidence on a picture which can be scrutinized again and again and again. Whereas you can imagine taking a visual acuity, reading off the chart, you can have good days, you can have bad days. So there's always some variability in that. Now, so with the regulatory agencies, FDA and EMA, so if they're keen on functional measures, which they are turning around on a little bit now, with especially in macular degeneration, they're starting to accept more structural measures. But for now, they are still primarily focused on functional measures. So the question arises, which functional measure is best? So I'm just going to quickly go over some of the functional tests, and those that have been on a clinical trial or have been for eye tests will be familiar with most of these tests. So the first one is just the plain old visual acuity. So it's reading the chart. And on the right-hand side is the addition of a dark filter, it's like a partial sunglasses filter, which then gives you a low luminance visual acuity. Now, as we know in choroideremia, though, it's the peripheral vision that's mostly affected, and especially in the early to mid stages. And even right up until the very late stages, you often find visual acuity can be very good, if not excellent, or normal. So we have regularly see patients with very advanced choroideremia who are still 20/20 on the chart. So that's clearly not measuring the stage of the disease very well. There is microperimetry, which is basically like a visual field test, but a much more advanced one that's come out more recently in the last 10 or 15 years or so. And this incorporates a visual field test, so the flashing dot test, but with a fundus camera, which has an automatic eye tracking. So it really takes care of some of the errors we were finding with the old visual fields, that your eye could wander and you wouldn't know if the patient's eyes were wandering all over the place, and that was giving you a false reading. This one will actually log where the patient is looking and compensate for that. So this is just a kind of simulation of what microperimetry looks like, which a colleague of mine at Oxford, Laura Taylor, made. So this is basically what patients go through. They look at within the machine, they see a target like this red circle, and periodically flashing lights will come up at different intensities. And each time they come up, the patient will press the button. And at the end of it, you get a heat map here, which is the patient's retina with an overlay of the results of that test. And what this then gives you is a retinal sensitivity map, which shows the areas that are very sensitive and normal retina and areas that are non-functioning. So it's quite intuitive here that the black spots are where the patient didn't respond, there's no functioning retina, and the green and spots are where it's pretty much normal. There's contrast sensitivity, which again, you might have seen, which is very similar to the visual acuity chart, but the contrast reduces. And the theory is that this can be a more sensitive measure of photoreceptors, because it doesn't measure just the photoreceptors, it also measures the response further down towards the visual pathways and going to the brain. It's a bit more sensitive to the entire visual pathway. So the theory is, in earlier disease, perhaps the photoreceptors are functioning well, but the rest of the pathway might be affected earlier. They might see changes in contrast sensitivity before you see changes in normal visual acuity. And there are different types of contrast sensitivity, ones where there's just a letter chart and ones where there are gratings of ever finer gratings and less contrast gratings until a patient doesn't see them anymore. There's color vision tests, which basically in the retina, if you know the anatomy of the retina is made from cones and rods. As we know from choroideremia, rods are primarily affected, but cones are affected later on. And so this is schematic of what cones actually look like under very high magnification. And in the periphery, you've got a mix of cones and rods. But the rods are generally of one type. The cones tend to be categorized into three different types: the S, the M, and L cones, so short and medium and long wavelength cones. And each one detects different colors, or is most sensitive to different colors. So S being blue, short wavelength, medium being green, and L red being long. Now, whilst in choroideremia is mainly the rods that are affected first, once the rods are affected, the cones lose their support from the rods. You kind of need functioning rods to support cones. And so a little bit later on, your cones start getting affected. but they can be affected in different ways. You have different numbers of S, M, and L cones, and generally you have far fewer of the blue, the S cones. And it's thought that the S cones are most fragile and affected earlier. So this could manifest as color vision defects, and you often see choroideremia patients describe the color being different. And again, there's lots of different tests that you have for color vision. If any of you have been through this before, the one on the left takes about 2 hours and it's incredibly annoying. But the other tests, more computerized ones, have been developed that make things a bit more streamlined. So that's just a quick snapshot of the many different tests. So there are many, many different functional tests. And it's widely accepted in the community amongst clinicians that the visual acuity is a really poor indicator of how bad the disease is, particularly in choroideremia, where you have a really preserved island of vision in the center until very late on. So my goal is generally to show to find out which functional measure is best and to hopefully rank them in order of which is best, using all the past data that we've collected from all 3 or 4 trials that we've run here in Oxford. So the key question I wanted to ask was how do outcome measures rank in terms of how well they detect changes in the disease? So first, before we answer that question, I had to define how to define the disease state in the first place. How do you define whether someone's got early disease or someone's got late disease? And the best way we have of doing that, well, probably the best way, is actually a structural measure, which is autofluorescence. Now, autofluorescence is it's an imaging modality, so it's a standard test that most people undergo at the hospital, and it's part of the OCT sort of regime. So you normally have an OCT test, photography, but also an autofluorescence where a very bright light shines on you. And what happens here is that in the normal functioning of the retina, you have a granular material called lipofuscin, which is a very fine sort of granular material, which is a byproduct of the photoreceptors. And it gets deposited in the RPE at the very back of the eye here. So that just kind of circles, they're very small granules, almost kind of waste products from the normal functioning of photoreceptors. And this lipofuscin has the really convenient property that it fluoresces when it's excited by light of a very particular wavelength. And fundus autofluorescence, which is the full name, detects this excitation of light, this fluorescence. And this is essentially what you get. So you've probably seen these sorts of images. This is a blue autofluorescence. So it shines the blue light of a very particular wavelength, and the retina shines right back at you. But it shines in a very particular way where actually the dark areas in the surrounding this island you see are non-functioning, and the really bright areas are actually the functioning retina here. So the bits that have this lipofuscin. And when you get compromised retina, you tend to get lipofuscin shining even brighter. So you get this anomaly where you have a normal retina, which is reasonably dark. It doesn't fluoresce very much. But when it gets stressed, it starts to glow brighter and brighter until it goes completely, it's destroyed that part of the retina, and then it goes dark again. So you can kind of see here the functioning area of retina is very intuitive where it is there. So if we look at just a couple of autofluorescence images, this is a baseline image of a patient, and you can actually measure the area of that autofluorescence. And then you can measure it again here. We've got it 3 months later. It's difficult to see much of a change over 3 months. And this is part of why structural measures possibly have not been favored in the past, because it's very difficult to measure changes after 3, 6, maybe even 12 months. But if you look at long term, over the 5 years that we've been running the choroideremia trials in Oxford, we can see here that we've gone from baseline to year 1, year 2, year 3, year 4, year 5, and you can clearly see that the island's shrinking. And this is a completely reliable measure that as the disease progresses, this island definitely always shrinks. There's never any doubt that this is a good measure of the disease. So this is the kind of gold standard, the accurate measure of the disease that we use to as a surrogate measure of the disease severity. And this is what I use to basically perform this analysis of ranking functional measures against this gold standard here. So the idea here is basically to have a plot. You gather data, you make a plot of your gold standard, the fundus autofluorescence, or another name would be the disease severity, versus your visual acuity on the x-axis. You gain all the data points and you fit a line. And according to several metrics that you can get, which is your p-values and your r-squared, how well this line fits the data, you can tell whether visual acuity follows that disease state, the fundus autofluorescence, well or badly. And that gives you a metric of whether visual acuity is actually a good measure, whether it follows the disease progression well or badly. So you can do this with all of those functional metrics. So visual acuity, you've got color vision, you've got low luminance visual acuity, and microperimetry, and you can rank them against each other. But to do this, you do have to use a very particular type of statistical method called dominance analysis. You can't just do an analysis on one functional measure and compare it directly to another. You kind of have to build a model with all the functional measures so that you can compare like for like. So dominance analysis has been used in a lot of different areas, but as far as I can tell, not much in the medical field. This is used a lot in NFL, and I think it actually started with NFL. And it can be thought of as a cooperative game where you evaluate each player's contribution towards the overall game result. So you can imagine an equation here where you've got on your y-axis, you've got your team score, which is your ultimate sort of your metric, your surrogate, almost disease severity, or your overall team score is on the y-axis. And then you've got all your measures on the x-axis, which in NFL, they use the players. They actually put all the players on the x-axis. And the goal here is basically to show what each player's contribution was to that overall team score. And in the end, I don't know if they still do this, but they used to pay the NFL players according to what their contribution was to the team or to the overall team score. So if I take this statement again, dominance analysis can be thought of as a cooperative game where we evaluate each player's contribution towards the overall game result. We can subtly change it and say dominance analysis can be thought of as a cooperative game where we evaluate each examination type's contribution towards the overall fundus autofluorescent result. So we can apply the same principle instead of in NFL to eyes and put fundus autofluorescence on the y-axis and all the functional measures we measure on the x-axis and compare one with the other. So this is exactly what I did. I took data from the non-treated eye because we didn't want any interference from any potential treatment effects. We're kind of looking at the natural history of choroideremia to try and detect what the best measure might be in a future clinical trial. So we took the data from the non-treated eye of one of the trials, the phase 2 trial we performed here called Regenerate. And we had 12 patients, which we followed up for 5 years. So it ended up being a lot of data for not many patients, but it was a lot of data we collected over the period. And the end result was this plot, which ranks all of the measures we took in order here from the top to the bottom. Now, so if I just explain, the time being at the top is kind of understandable, which it basically makes the statement that the best correlation to the disease progressing is time. So if you just wait from 1 to 5 years, as your time goes up, the autofluorescence will reduce, the area of island will reduce. So that's got the best correlation between the two. But obviously that's not very useful for us to measure. Microperimetry was second here, and that is that map of the retinal sensitivities. That correlated very well with fundus autofluorescence. But all these error bars do overlap, so you can't really say definitively which is best. These are just kind of giving you hints. What you can say definitively from this, though, because the error bars don't overlap, is that right at the bottom is the visual acuity. That is ranked dead last as the most useful metric for monitoring patients or for correlating with the disease severity. So as we all know, that the phase 3 choroideremia trial here at Oxford failed because it didn't meet its primary endpoint of 15-letter gain in the treated eye. And this was really frustrating for us, as I'm sure for you guys, because we all know that visual acuity isn't the best measure. When you've got 20/20 vision or close to 20/20 vision, gaining 15 letters, which is an extra 3 lines, is going to be almost impossible, if not impossible. So you're starting with a point that you just know you're not going to be able to measure again. But this is kind of where the FDA really are keen on BCVA, because it's visual acuity, because it's pretty much what they use for glaucoma and lots of other clinical trials. But the difference is in those clinical trials, like macular degeneration and glaucoma, you do tend to get visual acuity that's affected in the earlier stages. And so measuring a gain in that makes sense, but it makes no sense in choroideremia at all. But the FDA and the EMA aren't experts on this topic, and so we do have to convince them of this. And the issue, which I spoke to Beth about before we started this, is that often these clinical trial endpoints are decided not really on scientific evidence, particularly, certainly not scientific evidence in choroideremia, but based on conversations had at conferences. They usually have what are called symposiums at things like ARVO, where they get experts in glaucoma. They might get an inherited retinal disease patient expert there, but they will talk about what the future should be for clinical trials, what the endpoint should be. And with a lack of evidence, that probably is the best way of doing it, but it doesn't show the characteristics of choroideremia. It doesn't show you that visual acuity is going to be a poor measure. And if you get a glaucoma expert on that panel who says, well, we do it in glaucoma, then that's kind of how it gets passed. That's how it gets forwarded to the FDA. And that's how that gets chosen as a primary endpoint. So our challenge really is to convince the FDA and the EMA that that isn't a great endpoint, and we're probably never going to have an approved treatment if we carry on with visual acuity. We need to move over to microperimetry, or possibly some of these other measures like Pelly-Robson, which is a contrast sensitivity function test, or low luminance visual acuity, which is actually much more useful than a normal visual acuity, and it takes the same amount of time as the same non-complicated sort of test. So that the idea behind this work was really to actually, rather than go to a conference and just have a talk about it, is to actually have some evidence base, is to publish this as a paper, which is under peer review now, so it should be hopefully published in the next month or two. And then hopefully experts can get together and actually say there is some published work that shows visual acuity is poor, not that we just know it from clinical experience that it's poor. They can actually point to a paper and say, we know it's poor because it's been proven here, and we should use a different metric. So more broadly though, the idea of dominance analysis, I think, could be used a little bit more in clinical trials. Generally, in a clinical trial setup, it's a requirement to actually do what's called a power calculation, which is a method of calculating how many patients you need to detect a treatment effect, a theoretical number of patients. So you do a sample size analysis to say, yeah, if the treatment effect is so large, then you need 30 patients in a clinical trial, and then they'll mandate that you should get that many patients enrolled in a clinical trial. If you need a minimum of 30 patients, there's no point in having 29 or 15 patients, because even if you have a treatment effect, that might have been a fluke. You can't statistically say it was a true treatment effect unless you have that minimum number. So we perform routinely a power calculation in the setup to have a preliminary number, sample size, and then you recruit based on that. You try to go for that number. And the proposal here is that we should have a dominance analysis at the clinical trial setup phase, or in a very similar way to determine the optimal outcome measure as well in order to detect treatment, not just a sample size calculation, but an outcome measure detection calculation as well. So that's hopefully the direction we're going. Well, from a future point of view, because I guess the big question is where are we going in Oxford? And I think I alluded to at the beginning to say that functional measures was preferred by the FDA and the EMA, but they are starting to move over to structural measures, probably mostly because of the recent macular degeneration studies. And so what we're looking at now, as well as functional measures, or in giving better advice on functional measures, we are looking at more specific targeted functional measures. In particular, we're looking at the autofluorescence again, but a very specific zone called the smooth zone within the autofluorescence. So during the trials, we noted that there were different kind of subtle effects within this island, this bright island. There was this mottled bright effect, but also at the very center here, you can see a dark area. And as I said before, when it's very healthy, it tends to be quite dark. When it starts to get stressed, that lipofuscin really starts to light up. And so you do get this two-stage where there's mottled part of the autofluorescence, but also the smooth zone of the autofluorescence. And we noticed in the clinical trials we had was that the smooth zone was actually more representative of potentially the treatment effect. It was more sensitive to showing that there was actually a treatment effect. So we are currently setting up a trial to do almost a natural history study of this specific smooth zone within the island, in the hope that we then have an optimal structural measure, but also an optimal functional measure, so we can present the FDA and the EMA with two options, or potentially use both options as primary and secondary endpoints. And this is hopefully going to give us or lead us to something which will actually detect the treatment effect that we know is there. And that is our hope for the future. So that is it from me. So again, I'd like to thank you. This is the actual lab group here in Oxford. We have a very fancy lab dinner every year that Professor MacLaren sets up at one of the colleges, and everyone gets dressed up in very strange outfits. But with that, thank you very much.
I'm not sure it's strange. It was beautiful, actually, quite stunning. Actually, the picture was, for those that could see it. This has been really very, very educational. I loved your analogies. The NFL, I noticed who you had. Certain players up there, are they?
I don't know anything. I picked them at random.
Oh, did you? Okay,
I don't know anything about NFL.
That explains it then. Let's see if there's any questions that come in. Sometimes it takes a little bit of time for a couple to come in. And as they do, I did have some people text me. And so let me just start with these. After the paper is reviewed by peers that you were referencing, and if they agree that new endpoints should be considered or best case adopted, how does that process work? And followed up with can patient advocacy groups like the CRF or individuals help that process move more efficiently?
Yeah, I mean, I definitely think so. I think the process probably is to get it out there, so it's a paper that can be referenced, because that's what's been lacking so far. Is you've got clinicians going to these symposiums and just going by their intuition or their experiences, but in different diseases. And that has not worked well for choroideremia. It would work potentially in other inherited diseases like Stargardt's disease. Even in RP, it potentially makes more sense to look at visual acuity and compare to glaucoma, although probably not so much. But in choroideremia, it really doesn't, because the island is so well defined. There really is this healthy island which doesn't degrade until the later stages, much later stages. So you just cannot use visual acuity at all in any shape or form. And so that's where I think getting the paper out there is of primary importance. That's where my focus has been, because as far as I know, there is no evidence to show what we all know, And this is going to be hopefully the first that there will be an evidence-based. After that, I suspect it would just be discussed at Arvo, again, in a symposium, but hopefully with these experts and hopefully Robert MacLaren and people like yourselves are in it, then it will be discussed in that context with people who actually know the disease as well as the evidence base behind it. I mean, I think it's becoming generally more widely known now simply because of the failure of these clinical trials. And there's been so many failures of clinical trials in all inherited retinal diseases. There's been a glut of them because of inappropriate endpoints. And so I think it is becoming is coming to the forefront because there's just so much time, emotions, money, everything invested in this. And to fail on endpoints is just gut-wrenching because we know that some of these treatments work and work really well. And it just completely stifles future research if we don't meet these endpoints. So it seems crazy to us that gene therapy isn't actually the hardest part of this whole thing. This futuristic sort of gene therapy we're talking about has kind of been done. It's the endpoints, the old endpoints that we've been around for centuries. That's what we're struggling with now.
Okay, the questions are starting to come in. Thank you for answering that. Here's the first one. I noticed that in an early slide, you noted 1 out of 50,000 males have CHM. As we have learned, many females now also can have impacts from CHM. Should we now start to include women as well in this statistic? Having large numbers of affected individuals may encourage more companies to pursue CHM as an indication, as it would make it less rare.
Yeah, certainly. Our focus has definitely been shifting a lot more towards female carriers, and definitely because they often have quite severe phenotypes as well. They often present with very similar sort of presentations as the male counterparts. So it's not the case that it's very mild in all cases. You definitely get some that suffer quite severely. So we've definitely shifted onto researching female patients as well in both RP and choroideremia. I guess the challenge, and it's not my area of expertise because there are definitely some people in our group that research female carriers and a little bit more than I do, but there is a lot more what we call heterogeneity. So there's a lot more variety in the presentation. So it does add a little bit more complication in that with clinical trials, you almost have to be a little bit ruthless in who you pick. You have to get someone with a very specific pattern, a very specific stage, someone who you know will show a gain or you suspect will show a gain and show it quite clearly. If you get patients that show slightly different patterns, something that you're not aware of how the trajectory is going to go, that makes it a little bit more risky in that endeavor. But there's a lot more natural history studies going on with the female carriers now, which will hopefully answer that question so that they could be included in these trials.
True. Here's the next one. Would these same endpoints be effective or appropriate to measure changes in CHM female carriers?
Yeah, I mean, again, I think it's the same measures that would be used, but the pattern that they would display could be different. Could be in severity, but also the overall pattern. So the retinal sensitivity can be slightly different. And we don't actually know yet. That's part of the issue, is that we need to do more natural history studies to show the actual pattern. And I know my colleague from Melbourne, Senna, who does a lot of the female carrier research, would probably have a better answer for that.
Great presentation. One of our members here is saying, very uplifting to know the endpoints are being reviewed and addressed in a way that the FDA will listen and understand. Persuading the FDA is not an easy thing. You got that right.
Yeah. And, you know, they can't be an expert, I guess, in all things, but we've got plenty of experts now. So I think, yeah, we just need to get their voices heard a little bit more.
Here's another question from a fellow mother friend of ours. Do you think CHMers, and I'm thinking here young patients, should multiply multiply every test annually every year in their follow-up with doctors. The process is not easy, so wondering what you think.
Oh, I mean, that's a difficult one. In terms of clinical advice on follow-ups, I wouldn't be able to tell you exactly. I think you just have to go with your healthcare professional on that one. I know it's quite an arduous thing, putting all drops in and going through all those testing. People on our trials go through absolute, 2 days of testing, and they're absolutely exhausted at the end of it. So young children, I can imagine, is probably a little bit of a nightmare trying to get them through all that. I don't have a clear answer for you on that, I'm afraid. I think it's such a rare disease. The more data we have, the better. But obviously, you have to weigh that up against, how traumatic it is for the children. Definitely.
And that was another question that follow-up came. Are these endpoints the same for young CHMers
Yeah.
as in children?
Yeah. So for very early or early stage disease, they can be slightly different. Some of our machines, like the microperimeter that we really rely on, and I'm trying to push that as more of a primary endpoint. It goes out a certain way out to 10 degrees radius on the retina, which is kind of adequate for mid-stage choroideremia patients where their islands are within that 10 degrees. But earlier stage patients, or younger patients, their retinas, their islands might be much larger, and it might go outside of what that machine's capable of. There is a new machine now called the Compass machine, which is the equivalent of the Maya, the microperimeter, but goes out to 30 degrees. So these things are always being developed, but that might be more suitable for younger patients. But hopefully, the FDA might come around to the last slide I showed, the smooth zone on the autofluorescence. That would be a really promising kind of structural endpoint. If they go for structural endpoints, that would be a really promising one, and that would be universal, that test, for younger and older patients.
Will you be incorporating any of the work done in previous trials, Night Star, with all the people that were involved in clinical trials?
I'm not exactly sure what the plan is for, because with the smooth zone analysis, that's our next stage. That is a colleague of mine who's setting up the ethics applications and processes to start that study up. And I'm not 100% sure what the inclusion criteria is for that yet. I think it's still in the design phase, so I have to see on that one.
Can you please read the 12 outcome measures on the bar graph side out loud? I could not read them. I think there were some acronyms that I was unfamiliar with.
Yeah, sorry. I purposely didn't really go through all of them. It was mainly the key ones, but I certainly can do, because they have long names. I couldn't fit them all in. So can you see my screen here?
Yes.
Okay, so microperimetry, as we know, is that retinal sensitivity test. Pelly-Robson is the contrast sensitivity, but the letter version where it gets less and less contrast as you go down. The CSF is also a contrast sensitivity, but it's the gratings, and it's a computerized version. And each of these, there's several CSFs. There's this SF10, which comes up 4th on the most important ranking here, and that's spatial frequency. So that's to do with how thick the bars are on the grating. So SF10 is actually the finest grating. And if we go down to number 7 on the ranking, you've got contrast sensitivity, so the grating, but the thickest one, the spatial frequency 1, then spatial frequency 4, spatial frequency 8. And the thinking is the spatial frequency 10, the really fine one, is most sensitive to changes in autofluorescence or changes in the disease state, because again, it has some contribution, not just of the photoreceptors and the degeneration of photoreceptors, but also the pathway, or the visual pathway, all the way to the brain. You need quite high brain function as well as the visual pathway function to process contrast sensitivity, especially when it's very dim, very low contrast. It uses higher function to do that. So it's a little bit more sensitive to disease changes, basically. Then we've got CCT, which is Cambridge color test, which again is a computerized test, which we have on our system, which is called a Metropsis system, and BCVA. So this we've got the three colors: deutan, protan, tritan, which is like kind of the red, green, blue, and then visual acuity, BCVA there. We do have quite a lot of other measures we did on this trial, which are things like ERG, and lots of other structural measures. But I wanted to keep this paper as just functional measures, just to get the message really clear, and hopefully might follow this up with a functional rank, a structural ranking paper as well in the future.
Do you see yourself working with any other institutions or groups to kind of coalesce, come together, and work within the process, you know, with the FDA, with the EMA, which is to redesign the way of evaluation? Does that make sense?
Yeah, I mean, I guess most of that is a little bit above me. This would probably be more Robert MacLaren sort of remit, is to having these discussions and strategies and collaborative efforts, whereas I'm more in the back room with my laptop, furiously typing away and analyzing. But yeah, I think Robert's always trying to collaborate, trying to convince other centers. Obviously, with the clinical trials, they're often multi-center trials, and we usually are heavily involved with those other centers to try and keep things as homogeneous and consistent as possible. But yeah, I mean, I'm hoping this paper will start something, a little bit of a conversation, and hopefully Robert can champion that at Arvo at some point.
Yeah, good. Hey, we have another question that just came in. How do these measures correlate with quality of life? At the end of the day, shouldn't we be using measures that affect the patient? Good question.
That's a great question. So the quality of life, which is what I alluded to at the beginning, was one of the measures. So your structural, functional, but also patient reported. So we call them PROMs, patient reported outcome measures. And those are something that the FDA would be really keen on, because it's directly sort of measuring patient benefit. The patient feels things are better. Then, and you can prove that with, say, a secondary measure of structure or function, then you pretty much home run on that. I guess the issue is that the questionnaires that we have at the moment have probably not been quite adequate. Questionnaires are often designed to be generic for eye diseases. If not generic, they then hone in on inherited retinal diseases, but not specifically choroideremia. And so it's very difficult to get a really good, accurate report, a PROMS questionnaire, basically, that really hones in on what the patient experiences. That has definitely changed a little bit recently. So there are quite a few questionnaires out there. Difficulty is always validating them. When a new questionnaire comes out, it has to be validated against something. Getting enough patients in to validate something, that's quite a large study involved in that. But that is starting to happen. And if we could do that, then that would probably be the ultimate way of doing it, especially not by itself, but having a combination of that with structure or function to show they all corroborate with each other. But the questionnaire data is inherently quite noisy. You'll have one day where the patient might feel that they're not so good, and then another day where they feel that everything's fantastic. And so that will inherently create quite a lot of variability in that, and it's quite a challenge to get rid of that noise in the data. I guess doing it more often, you could do it at home, so you have lots more data points and you smooth out that noise. So that's always a possibility, but it is a difficult one, and it goes a little bit, I guess, inherently against what most clinicians want to do. They want something where they can look at it, look at a picture and say, yes, there's been an effect there. So there might be a little bit of a mindset change in that as well. But it's definitely going that way.
Oh, here comes another question. The FDA tends to be averse to multiple endpoints. They ask for corrections for multiple hypothesis testing, et cetera. Do you think there's value in testing more than one of these, especially if a treatment shows improvement in multiple ways?
Again, great question. It is a little bit of a subtle one in that the primary endpoint, yes, it's a single measure. That's the one they care about. You meet it or you don't, you pass, you fail. The secondary endpoints tend to be there, and you think, why are they there then if they don't even look at that? They are secondary, partly because they're exploratory. If it's a phase 2, didn't meet its primary endpoint, we've got a load of secondary endpoints. We then can go to phase 3, shifting to that secondary endpoint, make it a primary endpoint. But the other reason is if the primary endpoint is ambiguous, if it's not completely showing a treatment effect or is not statistically significant, but it's clearly showing an effect, it just doesn't meet the significance, there is an argument, and I don't know if there's been precedence to this, but there is an argument for going to the FDA if your secondary endpoint is showing a very clear statistically significant gain, that you can argue, look, it's a secondary endpoint, but it's really showing gain. The primary one is also showing a gain. It just didn't quite meet it. So I think that's kind of how they do it. You're absolutely right. If you have 2 primary endpoints, then your multiple hypothesis means your sample size, you effectively halved your sample size. So you've got double the work to prove that it was a real effect. So often that isn't the way to go. There's an argument for perhaps combining, in the same way you combine height and weight to make one index of BMI, and you can make BMI your endpoint. You can do that. That doesn't involve multiple hypothesis compensation for multiple hypothesis. BMI is one index. That would just be a single measure. So you could potentially do that in eye research as well. It's just tricky, I think, and not intuitive to people. Again, clinicians like what they can interpret easily.
Thank you. We have a couple more. Do you have a few more minutes?
Of course, yeah.
Yeah, thank you. Going back to the quality of life, have you incorporated any facial recognition tests into your measurement endpoints? That is the number one thing I hear from patients, is that they want to continue to be able to see their loved ones' faces. Being able to document this patient-centered outcome would be extremely valuable. Ooh, that's great.
That is great, yeah. And I think that was one of the issues with the questionnaire we used previously on all the trials, was the NFQ 25, I think it was called. And it was a pretty awful questionnaire. It didn't really have anything specific. There was one section in it that was more specific and asked about low lighting conditions and troubles patients had in low lighting conditions. But it was literally 3 or 4 questions out of the 25 questions that were specific to low light conditions. Don't think it had anything to do with facial recognition. It was things like, do you have trouble, or do you have trouble tying your own shoelaces and things like that? Very specific questions where you say yes or no, and it didn't give you an option for it depends. So that's always been a bit of an issue, those ones. But I think the newer ones that have come online are much more targeted. I don't know if they've got the facial recognition. That would be a great question. I'd have to go and look. We haven't quite started using them yet, but I know in future trials we are starting to use one called, I think it's the Michigan questionnaire, Michigan something questionnaire, which is quite probing.
In recent meetings with the FDA, CBER and CDER Victoria, had 2 large rare patient advocate conferences, NORD and Bio Summits. Those directors noted that there is a real value in patient advocates in helping push for more effective and patient valued, if not why they have the CRC reps there.
Yeah, I mean, I think you probably know more than I do about that side of things, and then definitely if there is scope to influence them on your side, yeah, I would. I'm sure you're already doing it, but I
do quite a bit.
Yeah, if we can move away from visual acuity, I think all the better for these trials.
Mm-hmm. I think that's one of the things that the CRF does really well is we try we do it very well. And as we try to extend our network and advocate a lot by Eric Hartman, Director of Advocacy, as we said earlier, Kathy Wagner, our Executive Director, we try to get really involved at the foundational levels. And it is very exciting to hear that things are looking at, they're reevaluating endpoints in clinical trials because it means a lot.
Yeah, absolutely. It was incredibly frustrating, and definitely I know from Robert's point of view. So yeah, it's definitely being looked at actively now, and in the ethics stage of starting the study.
Well, we just want to say thank you. I think one of our mothers, patients, or attendees here have noted, it is very encouraging, and these types of conferences and certainly webinars bring hope. It encourages us to keep working hard, keeping our foot on the dial, just to keep going.
Yeah, absolutely. It's when I first started this job, I was amazed at the impact it has, having the patient advocacy groups and things like that. The patients themselves, well, at least one or two, our microperimeter was actually paid for via a charity that fundraised for it. Prior to that, we never had anything for that. And so that's kicked off the entire field of microperimetry in choroideremia for us. So yeah, absolutely important, absolutely vital.
Well, thank you for joining us today.
Thank you very much.
Here's another just a comment coming in. Kathy, thank you for your excellent presentation. And others are saying, thank you, thank you, thank you.
No
So
problem.
on behalf of us, thank you very much.
Thank you very much for having me.
We'll hopefully have you again with more information.
I'll
Okay.
keep you updated. Yeah.
Thanks everybody for joining us, and we'll see you soon, and have a great, awesome day. Bye-bye.